Schramm has served as chairman of biochemistry here since 1967 and is truly one of our institution's visionary scientific leaders. Vern was also a decisive voice in our institution's revamping in the mid-90s of our clinical research infrastructure and the designation of precious space in the medical school for our CRC. More recently, I recall the day when Vern came to meet me in the planning of the CTSA, came to my office, looked out the window at the bare steel structure of this building, which was still under construction. He looked out, smiled, and said, "This is the time we should be planning the next building." I think we are. As one of our premier investigators, Vern has a stellar scientific record I don't have the time to recount this morning. Suffice to say that he was elected to the National Academy of Sciences in 2007. In its award, the Academy cited his analysis of the transition state structure of purine nucleoside phosphorylase and studies of purine salvage and protozoan parasites that have led to the development of new therapies for human diseases. Vern's research has solved the transition state structures of drug target enzymes and had led to the synthesis of some of the most powerful known enzymatic inhibitors. Dr. Schramm holds the Ruth Merns Chair in Biochemistry, and now he'll give us a little bit of an overview of this work and perhaps a glimpse into the future that he's so good at perceiving. Vern?
Thank you very much, Harry, and it's a great pleasure to be here. It's a hard act to follow to hear this history of the development of biomedical sciences in translational medicine. We've been lucky enough to be part of this in a way starting from physics and chemistry, which you mentioned at the start of your talk. And I must say that life is full of serendipity, and we're certainly victims of that in our laboratory. So our interest for many years has been simply how do enzymes work and what is the nature of the most critical moment in catalysis, that moment when the bond is being broken to convert substrates to products. We realized that this might be of interest beyond chemistry because theoretically, if one knows that structure at the instant of bond breaking, one can make a mimic of that, and that will become the most powerful inhibitor that could possibly be made for a target. So we had that in the back of our minds from the outset. We never actually thought we would get there. We were simply trying to do chemistry. So I'm going to talk about how we do this, and then in 2, 3, and 4, talk about how we've been able to go beyond chemistry and really to do some translational applications of this theory. And finally, I should say that this is a powerful collaboration in chemistry with our friends in New Zealand, Industrial Research Limited in Lower Hutt, New Zealand. Don't ask me how we got there. But in any case, it's a true collaboration between clinicians here, biochemistry in my laboratory, and chemistry in New Zealand. So I'm only going to have one slide on the theory because it's too complicated to go too deeply. However, I should tell you that intrinsic kinetic isotope effects are the cause of substitution of a heavy atom like carbon-14 into a bond that's being broken by an enzyme. And it turns out in quantum chemistry that the isotope effect, which is simply the change in rate of the enzyme reaction because it has a heavy nucleus at one site in a molecule, turns out to be a measure of the vibrational environment of that atom at the instant of the transition state. So this is something that's been known for a long time in theoretical chemistry, and we've simply expanded that outward so that we make a family of molecules with heavy atom isotopes individually at a number of positions. So this is tough synthetic chemistry to make isotopically labeled compounds, which we do in our laboratory. We measure these isotope effects, and you can see they're small. This number, 1.026, is the ratio of reaction rate between the unlabeled molecule and the labeled molecule. It means that the labeled molecule with carbon-14 here reacts 2.6% slower. And that number, if we can measure it accurately, tells us the nature of the transition state. Combining these experimental measurements together with computational chemistry, quantum chemistry, that tells us where the electrons are on the van der Waals surface at the instant of the bond breaking, gives us then a blueprint that we can try to mimic in a stable chemical compound, and that will become an antibiotic. So there are only two forces in nature that lead to biological recognition: shape and electron distribution. There's nothing else. So this is the van der Waals surface of a molecule at the instant of its bond breaking at the transition state. If we know this, then we can try synthetic chemistry, make molecules that are similar to this, and then see whether or not they inhibit their targets. Our first target really comes from this interface between the patients and clinicians. And this comes from a study by Eloise Giblett. In 1975, she was doing blood typing and was doing blood typing on a little girl who had T-cell disease, T-cell immune deficiency. And what Giblett found was that this patient was genetically deficient in purine nucleoside phosphorylase, PNP we'll call it here. And what happened is that these individuals that have this genetic deficiency are perfectly normal at birth, but by the time they're 1 or 2 years old, they start to lose their T cells, and by the time they're 3 or 4, they're completely T cell immune deficient, which means that they're going to die of viral infections if they don't get an appropriate bone marrow transplant. There are about 50 of these patients who have now been characterized biochemically, and we know exactly what's happening. And here's what happens. In normal human metabolism, the DNA, of course, from cells that are being replaced is always being degraded, and the Gs that come from DNA apoptotic cell turnover are released as deoxyguanosine. In us, that deoxyguanosine is removed immediately by purine nucleoside phosphorylase. If this is genetically deficient, then there's no escape for deoxyguanosine from cells, and it's converted into deoxy-GTP, one of the precursors for DNA. It turns out that uniquely in dividing human T cells, there's a pathway that accumulates deoxyguanosine, takes it forward to deoxy-GTP, that unbalances the nucleotide pool for DNA synthesis and triggers apoptotic death, specifically in dividing T cells, not B cells, not any other cell type. So this is a specific T cell disease driven by a single gene mutation. Immediately, the pharmaceutical industry recognized that knocking out that gene with a pharmaceutical agent would be good for T cell cancers, which are rapidly dividing T cells, and for autoimmune diseases, most of which are driven by T cell misrecognition of human cell types. When we started this project, there were already 35 patents in the literature from 7 major pharmaceutical companies that tried to target this enzyme and failed. We thought we might be able to attack this by transition state theory and to see what we could do to knock out this enzyme and recapitulate this in unwanted dividing T cells. So we started this project, and this is just a flow diagram for how we attack all of our problems now. We select a target enzyme. We used the bovine PNP simply because we could buy it from Sigma at the time instead of the human one, which wasn't available. Measure the isotope effects, do the quantum chemistry, get the electrostatic potential map, this electron cloud, do the chemistry, and then evaluate the compounds biologically. And this is what the process looks like at the end of our really our chemistry and physics analysis. If we look at the reaction for purine nucleoside phosphorylase, this is the reacting molecule, its substrate. And this is the electrostatic potential map for that molecule before it binds into the catalytic site of the enzyme. At the instant of the transition state, you can see it's very different in electron distribution and in some aspects the geometry from the reacting molecule. We can design molecules now that look like the transition state, and clearly these two molecules are more closely related to one another than either is to the reactant. And that's all we need to capture some of that transition state binding energy. Well, it turns out this compound is a 23 picomolar inhibitor of human PNP. That's orders of magnitude more powerful than anything pharmaceutical companies could come up with. This molecule binds 700,000 times tighter to the enzyme than its normal substrate does. And it's now in phase 2B clinical trial under FDA-approved special protocol assessment, meaning it's going to be it'll be FDA approved directly out of this phase 2B pivotal trial for cutaneous T-cell leukemia patients who are not responding to other therapies. So this is a great validation of this kind of approach, which had been tried by major industry and hadn't succeeded. So at the same time, of course, we were very interested to see whether or not these inhibition constants or affinity for the target enzymes were the same for the bovine, which we had used as our design target here, and for the human PNP, and they're close, a factor of 2 or so different. However, we worried about this factor of 2, and it raised the possibility that had been unconsidered in chemistry before, that two very closely related enzymes could possibly have different transition states. So we simply went back to repeat this. I should back up a minute and tell you this is an example from a phase 1, phase 2 clinical trial in a patient with T-cell leukemia. This is giving the compound, this compound, IV at time 0, and it's a biochemical experiment to see whether or not we can change the blood chemistry in the same way that happens in patients that are genetically deficient in this disorder. Single injection of the drug, the deoxyguanosine concentration, which is normally 0 in humans, jumps up to micromolar concentrations, continues to increase over this 5-day period of therapy. The concentration of deoxy-GTP inside of the cancer T cells go up 60-fold, 60 times above the normal. And this is the T cell burden that's reduced by about 80% in just 5 days of therapy of these agents. These agents are not metabolized in humans, they're not toxic, and they're orally available. So this is the possibility of a once-a-day oral therapy without side effects for an otherwise lethal cancer. So moving ahead, we thought maybe that we hadn't done the right thing, that it might be possible that the human enzyme might actually be a little bit different. So we just went to repeat our entire process after we'd cloned and overexpressed the human enzyme. This is the primary data set from the bovine PNP, where we're comparing kinetic isotope effects directly. And we'll just look at one. For bovine PNP, the carbon-14 isotope effect is 2.6% here, and it's 0.2 here, a factor of 10 different. This is the primary data set based on bond vibrational differences at the instant of the transition state. And clearly, this transition state for the human enzyme is different from our biological or our chemical model of the bovine enzyme. So we made a mistake. We should have started with the human one first, but we were very happy to find this before anyone else did, in fact. So we went back and compared now our electrostatic potential models. This is the reactive inosine again. This is the transition state for the human PNP, and you can see it's very different what you saw before for the bovine. Here we have 3 angstrom or complete electronic separation between the leaving group and the ribocation at the transition state. We can mimic that by stretching out the 2 halves of our drug by putting a methylene bridge between the 2, incorporating this nitrogen charge closer, nitrogen cation charge closer to where it is in the actual transition state. You see what a nice match we have both in geometry and charge here. And this molecule is a 9 picomolar inhibitor. It binds to the human enzyme 4 million times tighter than the substrate does. So this is a record in this field for making powerful inhibitors. We try this experiment in the lab to see whether or not these kinds of compounds are going to be orally available. We simply starve a mouse overnight, put some of the drug on food and feed the food in the morning, and then measure the target to see whether or not we're inhibiting the target enzyme as a function of time. If we are, that will tell us that it's orally available. In other words, it gets through the gut, through the membranes, into the red cell where we're measuring the enzyme activity. And then we can also see how long it takes for the animal to clear the drug. In other words, what's the biological efficacy of the drug on the target? When we do this for this compound that we call DADMe-Immucillin-H, we find that the half-time of inhibition has an onset of 10 minutes. So it's clearly orally available. After this single oral dose, we see that the enzyme activity is 0, and then we change the time scale here. It takes 11.5 days to regain 50% of the activity, 11.5 days for a single dose. It turns out that that slope is the rate at which the mouse replaces its red blood cells. This is not loss of the drug is not lost. So it stays on the target for the lifetime of the red cell. So we call this achieving the ultimate goal in inhibitor design. Single oral dose goes on to the target and stays there until the cell is replaced. as one of my colleagues said, if you make inhibitors that are better than this, you'll simply be wasting NIH money. This is exactly the same experiment. This compound is now in clinical trials for autoimmune disease, and this is exactly the same trial that we did in the mouse, but done in 41 normal human volunteers, college students in New Jersey, to be precise. By BioCryst Corporation and Roche Pharmaceuticals are now developing this drug. So the object of this experiment was to see what the appropriate dose would be to, the appropriate minimum dose would be to get complete inhibition. And they made a little mistake because every dose is too big. Even at 0.5 milligram per kilogram, you can see that the purine nucleoside activity in human blood drops to 0, slowly recovers. This slope is 120 days. This is the lifetime of red cells in human. I just want to emphasize what a dose of 5 milligram per kilogram is. I don't know if any of you take your 81 milligrams of aspirin in the morning to prevent your heart attack. This is an 81 milligram tablet. 0.5 milligram per kilogram is about 1/3 of this tablet. So these are very powerful drugs. So this compound is now in clinical trials for psoriasis as the lead indication for T-cell autoimmune disease. They include inflammatory bowel disease, rheumatoid arthritis, multiple sclerosis, juvenile insulin-dependent diabetes, Graves' disease. There are 70 different T-cell diseases that are caused by overactive T-cells acting on human tissues, and we hope that this is going to be a lead indication with a great opportunity to do good for large numbers of patients. So these were both the same target, basically, purine nucleoside phosphorylase, and we'd like to extend this to other targets. And there's been a lot of talk about epigenetic control, and we are really just starting to learn about this now in terms of regulation both by DNA methylation and by changes in histones for control of gene expression in human. Epigenetics is really what makes a cell type a cell type. Of course, all cells have the same DNA, same gene structure, but that structure is regulated by epigenetic control. And I put epigenetic here. In fact, I just put this here yesterday when I was making this slide, because we didn't start off by trying to do epigenetic control, and this is a serendipity that discovery that we came from knowing other pathways of cancer metabolism. There's this drug called difluoromethyl-ornithine, or DFMO, that's been around for more than 20 years, and it's FDA approved for certain indications and certain kinds of cancers, but it's never been widely used. It's not a good drug. It turns out that you need to take 8 grams a day of that drug, and if you're taking enough to kill your cancer, you also go deaf during the period when you're taking the drug. Not a good drug. Our idea was to take another enzyme out of this same pathway, the polyamine pathway, and block that and cause accumulation of an intermediate, and that intermediate would control the pathway and prevent its end product formation in the same way that blocking the first step in the pathway would do. We knew blocking methylthioadenosine phosphorylase, an enzyme we now call MTAP, would do that, and it would also prevent the recycling of S-adenosylmethionine, a critical precursor for many pathways in human cancer metabolism, normal metabolism for that matter, including DNA methylation, which is a major pathway of epigenetic control. So this just came from Tuesday's Science Times in the New York Times. This is one of the chromosomes in humans, chromosome 22, and the bars represent different levels of DNA methylation at CPG islands, or the CPG sequence, which are not evenly distributed, but they're distributed in a regular way, in a regulatory way around the chromosome. 22 is one of the smallest chromosomes. This represents about 2% of the total DNA in humans. And what we're looking at here is the difference in different human tissues. For example, muscle, liver, heart, etc. And they all have different methylation patterns. methylation patterns are what make a tissue a tissue. It also is what makes a cancer cell a cancer cell. We thought that if we could block this methylation pattern, it might disrupt the normal development of aggressive cancers and be useful in cancer therapy. So we've gone through the same procedure I talked about before, kinetic isotope effects, solving the transition state structure, making inhibitors. We have inhibitors to 10 picomolar, very powerful inhibitors. We've selected this one for development. We call it methyl-DADMe-Immucillin-A because it's orally available. And we screened this compound against human cancer cells in collaboration with our cancer center. And we find that head and neck cancers in culture are susceptible to this treatment, and we can grow those in immune-deficient mice. So this is a 28-day after injection of FaDu human head and neck tumors into the dorsum of the hind foot of a mouse. And if we treat with optimal doses, we can prevent the growth of the tumor. So this is just a graph showing the growth of the tumor in untreated and in treated mice. If we take optimally treated mice off this therapy, the cancer doesn't regrow, so we're killing the cancers irreversibly. There's no toxicity in this treatment, either by weight gain or gut histology. So we think this might be an epigenetic tool to get at human cancers. We've now tried other models as well, and we find that human lung, prostate, cervical, and head and neck cancers in mouse models all respond to this kind of therapy. And if we take tumor pieces out and look at the DNA methylation patterns, they're clearly changed dramatically because of this treatment. So the treatment appears to be disrupting DNA methylation that's required for tumors to thrive in an animal and undergo the progressive genetic changes that lead to cancer aggression. Fortunately for us, we're not epigenetic scientists, and if we were, we probably would never have started this, because it turns out that if you knock out this gene, MTAP, in mice, it's embryonic lethal day 8. However, we know that if you give it now to adult mice, nothing happens. Their epigenome is already developed in all of the terminally developed tissues, so we're not disrupting them. We're only disrupting tissues that are incompletely developed. We're doing the same kind of thing now with other targets, quorum sensing in bacteria, which is required for virulence. If you knock out quorum sensing genes, you prevent some of the virulent properties of these kinds of human pathogens. We've done transition state structures. We have inhibitors in this series to 47 femtomolar. That's 91 million times tighter than the natural substrate binds. We can knock out quorum sensing molecules. This is in Vibrio cholera with these kinds of molecules. And we can do this without affecting cell growth. So we're putting no selective pressure on the bacterium, so there's no way that the bacterium can escape this kind of drug therapy. So we call this our everlasting antibiotic program. So finally, transition state theory and chemistry create some powerful inhibitors. We have inhibitors for several indications. We have many more that we're trying to do development. We've been well supported by the NIH and other agencies. These are the people in the lab. Thank you for your attention.